Hello everyone, my name is Sercan and I am a PhD candidate at Bezmialem Vakif University in Istanbul. I am going to talk about encapsulation of CCHFV-NP mRNA with PLGA, potential utility of PLGA mRNAs. PLGA, polylacticoglycolic acid, is nanoparticle carrier system. CCHFV is Crimean-Congo hemorrhagic fever virus. NP is also a nucleocapsid protein of CCHFV structural protein. Extensive research into CCHFV immunology, virology, and biology is global priority. mRNAs have garnered attention for their advantages over conventional gene transfer methods. Formulation technologies for molecular encapsulation have also advanced significantly. In the study, we employed PLGA to formulate modified mRNA containing the unoptimized S segment of the CCHFV capsid strains. This study aims to analyze NP mRNA encapsulation structures created with PLGA stored at room temperature using DLS, dynamic light scattering. This study aims to also determine the amount of the encapsulated mRNA and clarify its release rates. The main objective is to assess specific antibody responses in mouse serum after storing the prepared formulated NP mRNA and naked NP mRNA at room temperature. This is done by immunizing mice and using an in-house ELISA. When it comes to the materials and methods, our first goal is to create CCHFV-NP mRNA, a method called in vitro transcription, IVT transcription method. This happens in this tube. While making it, we use uracil and pseudouridine in equal amounts, like a 50-50 mix. We start by treating the mRNA with DNase. After that, we clean it up using lithium chloride. Then we check the mRNA using a nanodrop and agar shell. The B figure first illustrates the preparation of PLGA. After dissolving PLGA in acetone, it is slowly added drop by drop into DPBS using a syringe. Acetone is evaporated overnight. Then mRNA is mixed with PLGA in a 1:8 ratio. This mixture is vortexed for 10-15 minutes and at 10-second intervals and kept on ice. To confirm the encapsulation of the prepared formulation, agarogel electrophoresis is performed. The encapsulation rate is calculated using nanodrop and ribogreen. The product is left at room temperature for 60 days and measured with DLS. We do release tests for the encapsulated mRNA and check naked mRNA stability over 24 hours at room temperature using nanodrop. Mice were immunized 3 times with naked mRNA and formulated products that had been incubated at room temperature. Following immunization, blood samples are collected from the immunized mice. The sera are separated from these blood samples. NP-specific IgG antibodies are screened in the sera using an in-house ELISA. For this purpose, ELISA plates are coated with in-house recombinant NP. Most sera are used as the primary antibodies. Secondary antibodies is most specific anti-IgG HRP conjugated antibody. After the addition of the substrate, the reactions are stopped and the results are measured. Absorbance for 150 graphs are then generated. The initial results include visual representation of the IVT mRNA generated in Figure A. In Figure B, there are agarogel images of samples taken at different stages of mRNA production. The poly-A tail was enzymatically added subsequently. Therefore, there was an approximate increase of 150 bases after the poly-A reaction. In figure C, NP mRNA with a concentration of 500 nanogram per microliter is incubated at room temperature for 24 hours. Nanodrop measurements are taken at specific time intervals. It can be observed that approximately 2 hours later, degradation begins to increase. After 24 hours, the degradation is more than half of initial concentration. However, it is unknown whether the remaining mRNA is intact or not. Degradation may have started from the end, maybe. In figure D, we figure out that about 53.3% of the NP mRNA was encapsulated when we used ribonucleic acid nanodrop. But the encapsulated NP mRNA with PLGA mixture and naked mRNA were loaded onto agar gel at some concentration in the gel image. Despite the formulation product containing mRNA at some concentration, the band profile appears less prominent. The reason for this is the encapsulation of a portion of the mRNA. In figure E, the release of encapsulated mRNA was measured using nanodrop. The percentage ratio has been presented graphically. The calculation indicates the release of approximately 30% of the mRNA from its encapsulation structure after 24 hours. Figure 2 shows that we measured the formulated mRNA using DLS at room temperature for 60 days. This represents the encapsulation structure, which can be observed to remain stable over the course of 60 days. However, these results do not provide information about the fate of mRNA. Conclusion of this result is encapsulation stability at room temperature, approximately 100 nanometer size distribution by intensity, number, and volume. Figure 3 contains the results of the main objective of this study. I would like to read the title of this results. CCHFV-NP specific antibodies are triggered by stored NP mRNA at room temperature. It sounds interesting, yes. Here we are looking at figure A. In this figure, 25 micrograms naked mRNA is freshly produced and kept at room temperature for up to 24 hours. Mice are immunized 3 times with mRNA having a fresh starting concentration of 25 micrograms each time. The results are analyzed to detect NP-specific antibodies using ELISA. It is observed that fresh naked mRNA can immunize mice starting from the first immunization, and mice can produce NP-specific antibodies. The effect of the first immunization disappears after 4 hours at room temperature. Similarly, after 4 hours, mice cannot achieve significant antibody production in the second immunization. However, the third immunization support the production of the desired antibodies in mice up to 24 hours. In figure B, IVT mRNA is prepared at a concentration of 25 micrograms, formulated with PLGA, and injection are started intramuscularly. The formulation are kept at room temperature, continuing up to 45th day in formulation group. Mice are immunized 3 times with IVT mRNA formulation with PLGA, having a fresh starting concentration of 25 micrograms each time. The results are analyzed to detect NP-specific antibodies using ELISA. The fresh formulation group obtains high absorbance values significantly higher than the naked immunization group. However, it appears that the first immunization in the naked group has produced slightly more antibodies, while the second immunization in the naked group is not very effective, the formulation group has provided more significant increases in absorbance at the second immunization. Significant antibody responses are detected until the second day when left at room temperature, but the second immunization on the second day did not induce a significant antibodies response. Immunization with the formulation left at room temperature for 7 days did not generate a significant antibody response in mice during the first immunization. By the 38th day, first two immunization no longer elicited an antibody response. Furthermore, after 45 days of incubation at room temperature, the formulation reduced the antibody response after the third immunization. Thus, it has been observed that even after 45 days of incubation at room temperature, the formulation group generated a significant antibody response following the third immunization. In conclusion, to summarize the study's findings, after 2 hours at room temperature, naked NP mRNA degrades, yes, but it induces specific antibodies within 24 hours after the third immunization in mice. PLGA mRNAs encapsulated at approximately 53.3 plus or minus 2.75%, but approximately 30% is released after 24 hours. The ELIS results show stability of encapsulation structure for 60 days at room temperature, but offer no insight into mRNA's fate. After the third immunization in mice, formulated NP mRNA continues to induce antibodies after 45 days at room temperature, but its efficacy diminishes with time. In summary, PLGA mRNAs proves more stable and durable at room temperature than naked mRNA, while still capable of eliciting specific antibody response in mice. Thank you for your attention. This study has been supported by Bezmialem Vakif University. You can write your question in the comment section. Thank you again.